Okay, so welcome to this next video in which we are discussing endocytosis. Okay, so at the moment we are looking at the way in which endocytic motifs were discovered, and they were discovered by Brown and Goldstein, who were investigating familial hypercholesterolemia. Okay, so at the moment we're just discussing what an LDL particle is. So an LDL particle has this sort of ring of phospholipids where the phosphate heads of the phospholipids, which are polar, they face the outer surface of this sphere of phospholipids, okay? And the hydrophobic tails, which are these long-chain carboxylic acids, which are esterified to the first and second hydroxyl groups of the glycerol backbone of the phospholipid, those all face inwards and they interact nicely with each other. Now, that is the lipid part of a lipoprotein. However, we now need to discuss the protein part of the lipoprotein. So you don't just have phospholipids in this little particle here. What you also have is some proteins. Okay, so here let's have this as our protein. And this protein is apolipoprotein B1. So apoprotein B1 actually, not apolipoprotein B1. Apoprotein B1. B100 rather. Okay, so apoprotein B1, this is, in fact I think it is apolipoprotein B1. Anyway, it's often abbreviated to apo B1. B100 rather. Apo B100, right. Sorry about this. The protein that you find in low-density lipoprotein particles is a protein known as apolipoprotein B100. Yes, it's apolipoprotein. I'm pretty confident of that. Apolipoprotein B100. Okay, often abbreviated to Apo B100. Okay, right. And this is the protein that is inserted with the phospholipids in this LDL particle. So you don't just have the phospholipids, you also have this apolipoprotein B100, or Apo B100. Okay, and that is what makes this an lipoprotein. The fact that you have this lipid, the lipids in this particle, and also the protein, so it's a lipoprotein. Now, the low-density lipoprotein, why is it known as a low density? Well, the density refers to how concentrated the protein is within the lipids. So, a certain fraction of the molecules in this lipoprotein will be lipid molecules, and a certain fraction will be protein molecules. Now, if the protein concentration is very high, then that will be known as a high-density lipoprotein. The protein concentration in LDL is very low, so you have mainly lipid molecules and not very many proteins, which is why it's a low-density lipoprotein. So it's referring to the density of this ApoB100 protein. Okay, right. So, in these lipoproteins, you don't just have phospholipid and ApoB100 proteins. Instead, you also have other lipidic molecules as well. So you'll have maybe some triglycerides in there, which is just a glycerol molecule with 3 fatty acids esterified to it. So this is a triglyceride here. Okay, so a glycerol molecule with 3 fatty acids bound to it. Okay, so you might have some triglycerides in there. You might have some free fatty acids, i.e. fatty acids which aren't esterified to the glycerol molecule. And also, you'll have cholesterol in this, in these particles. So you'll also have cholesterol. Okay, and that's important. So, basically, these low-density lipoproteins, then, they are a way of transporting lipidic molecules around the body within the bloodstream, and they contain cholesterol. So, basically, what we're going to look at is these people with familial hypercholesterolemia. What they are going to have mutations in is the Ldl receptor. Now, let me explain what the Ldl receptor does. So, these LDL molecules, they are in the bloodstream. Now they need to be taken out of the bloodstream. So what takes them out of the bloodstream? They were put into the bloodstream, but there's no point putting something into the bloodstream unless it's going somewhere. So they need to be taken out of the bloodstream. And LDL molecules are generally taken out of the bloodstream by liver cells, so by hepatocytes. Now how does this work? Well, basically, what usually happens is the hepatocyte will have an LDL receptor on it. So here is the hepatocyte membrane with the LDL receptor on it. The LDL molecule, or this particle, the LDL particle will come here, and we'll denote this LDL particle just as a pink blob from now on. So here comes our LDL particle. Okay, so this is the LDL particle. What then happens is that the LDL particle is going to be endocytosed by the hepatocyte. So let me show this. So if this is our hepatocyte here, what's going to happen is that you're going to endocytose this whole structure here. You're going to endocytose the LDL receptor, which is this molecule here. So this is the Ldl receptor, which I'll just refer to as LDLR. You're going to endocytose the whole LDL receptor with the LDL particle bound to it. So what's going to happen is an adapter protein is going to come and bind to an endocytic motif here. So here comes the adapter protein, which will bind to the endocytic motif, and it will only bind to it once the LDL is bound. Okay, so this horseshoe structure here, this is the adapter protein that's come and bound to our endocytic motif, which is in turquoise here. Okay, and now what will happen is you'll get the budding off of the vesicle. So if I draw this process happening, so you're going to get the budding off of the vesicle like so. Here's the vesicle that's being budded off. Okay, and the LDL receptor is still there, so I might have to draw it a little smaller now. So here's the LDL receptor, and it's got its LDL bound to it there. Okay, and as I've said in earlier videos, this process of budding off is dependent upon the protein clathrin. So clathrin will form a structure which pulls the membrane down into this bud here. Okay, and then finally you'll get this pinching off of this budded vesicle to actually get an endocytic vesicle. So a vesicle that's within the cytoplasm of the hepatocyte here. So here's our hepatocyte, which actually contains the LDL receptor and also bound to it the LDL particle. So here's our LDL particle bound to our LDL receptor now. Okay, and then what will happen is that it will process that LDL and remove the lipid contents of the LDL. So this is the mechanism by which LDL is removed from the bloodstream. It goes into hepatocytes via this endocytosis mechanism. So this here is an endocytic vesicle. So remember, an endocytic vesicle is the correct name for one of these structures that has been endocytosed, not an endosome. Do not use endosome. Endosome is something different. Okay, right. So, now, what happens then in familial hypercholesterolemia? Well, basically, these people have loss of function mutations in the Ldl receptor. Okay? Now, if the Ldl receptor is not functioning, then you won't be able to remove LDL from the blood, and therefore the LDL will remain in the blood, build up in the blood, and because there is cholesterol in the LDL, that will mean that the cholesterol level in your blood is going to go up. Hence, you get hypercholesterolemia. Okay, right. So, we have two genes for the Ldl receptor. So we'll have one on our chromosome that came from our mother, and we'll have one on our paternal chromosome. So every gene we have two copies of in the human genome, at least for chromosomes 1 to 22. Okay, so we have these two LDL genes. Now, so let's show these LDL genes as these boxes here. So this is one LDL gene, let's say that's the paternal one, and here's our maternal LDL gene. So these are the LDL gene. Right, so if we suffer loss of function mutation in one of them, then that means one of them is no longer going to function. So let's say it's this one here that no longer functions. Then what's going to happen? Well, okay, we're not going to produce as much LDL receptor because this one's no longer working, but we've still got one that's functioning, so we should still be able to endocytose some LDL out of the bloodstream, so hopefully we won't get too severe a symptom. This is known as a heterozygote. So if you've got one mutation in one of your genes for LDL receptors, then you are known as the heterozygote for the mutation, and you then get a milder phenotype than if you'd knocked out both. Generally, these people they do have hypercholesterolemia. Their bloodstream cholesterol level is too high, but it's generally only too high by 3 times. So they have 3 times the correct level of cholesterol within their blood. And this does predispose them to cardiovascular disease. Generally, they die of heart attack. So they die of myocardial infarction, MI for short, during middle age. So when they're middle-aged, Okay, so they have shorter life expectancies than most people, but it's not as bad as if you'd knocked out both. So if you knock out both genes, i.e. you are a homozygote and you have no functional LDL receptor, then your cholesterol level within the bloodstream is 10 times what it should be, okay? And these people die in childhood. So if you're a homozygote for loss of function mutations in LDL receptor, you die during childhood. So it's a more severe disease, basically. Okay, right. Now, there are different mutations which can constitute loss of function mutations in the LDL gene, and we'll discuss those in the next video.